immunology. He had an encyclopedic knowledge of the field. For 3 decades, he was chief editor of the Annual Review of Immunology, which under his guidance for 7 years was the most cited publication in biomedical science. He initiated and edited Fundamental Immunology, the most definitive reference in the field. He was, for a period, the editor of the journal Immunity. He was president of the American Association of Immunologists, the American Society for Clinical Investigation. Furthermore, he was chief of the Laboratory of Immunology from 1970 until his death. With his persistently positive attitude and his generosity, the laboratory was and is a resounding success. We all appreciate his ability to ask the most probing questions, yet make us feel we were receiving an accolade, a reflection of his modesty about his remarkable intellect. I and many of you in the audience have benefited from his generosity. By being nominated for an honor, award, or membership in an august society, actions where he appeared to have more enjoyment from our successes than from his own accolades. Bill's contributions to the research in the field of immunology are extraordinary. One of his first and important works was with the future Nobel laureate, Dr. Baruch Benacerraf and Ethan Shebeck, Ira Green, and Alan Osenthal, who showed that IR genes control the capacity of simple carrier molecules, such as mono and diamino polymers, to elicit immune responses to any hap genes conjugated to these carriers. He showed that NHC class II molecules played a critical role in the presentation of antigens by APCs to T cells, and that polymorphisms in class II determined the specificity of such control. His more recent seminal contribution with Maureen Howard was the discovery of IL-4, the elucidation of the molecular basis of IL-4 signal, dissection of molecular genetic control of the expression of IL-4 and of the Th2 cytokines, and the delineation of the general principle of positive cytokine feedback regulation of CD4 T cell effector polarization. For 4 decades, it has been my privilege to share an interest in the common gamma cytokines with Bill. Over the past 35 years, a major focus of my laboratory has been on the roles of IL-2 and IL-15 in the life and death of lymphocytes, and the implication of these observations for the immunotherapy of cancer. I have only a moment to touch upon IL-2, where with the Cossiuciano, we produced anti-TAC, diclizumab, Zyngrud, a monoclonal antibody that with Warren Leonard and Warner Breen was used to define and molecular clone the gene for IL-4 alpha, the discovery of CD25. So for decades, we have developed this antibody where in its humanized form for therapy, it was approved by the FDA for the prevention of organ allograft rejection and for the treatment of patients with multiple sclerosis. However, today I will focus on IL-15, a cytokine pivotal in the maintenance of NK cells and memory phenotype CD T cells. There will be 2 parts to my talk. The first, familiar to many of you, will be our efforts to bring to fruition IL-15 in combination therapy as an effective approach to the treatment of metastatic malignancy. The second part, which I have never presented, will be on disorders of IL-15 and its signaling JAK-STAT system in autoimmune diseases and T-cell malignancies, and our efforts to develop agents to take that into account and treat these disorders. So in terms of Janus, the Roman god, he looks both backwards and forwards into the future, as I will. And I will also give him both good and evil faces. IL-15 useful in therapy, IL-15 involved in disease pathogenesis. So IL-15 is shown here, is involved in an immunological synapse where antigen-presenting cells and IL-15 or alpha present its always marker IL-15 in yellow to cells of the beta and gamma chain of its tripartite receptor. I will tell you IL-15 when given to people has the capacity to dramatically increase the number and activation status of NK cells, a 30-fold increase in the number of conventional NK cells, and more than 300-fold increase in the number of CD56 right NK cells. And in rhesus macaques to increase the number of CD8 cells 100-fold to 100 per cubic millimeter. However, in each case, there is an injunction of a checkpoint. Thus, in order for IL-15 to reach its potential, it will have to be used in combination therapy that circumvents these checkpoints, that is rational, that is functioning in animal models, and is just now being translated into clinical trials. So IL-15, which as usual we discovered in serendipity while studying IL-2 receptor, is a 14-15 proton member of the 4-alpha helix bundle cytokine family. It and IL-2 share much. They both have as their receptors a heteroprimeric one with the gamma chain, also used by IL-4, 7, 9, and 21. And they share a beta chain, CD122, which I've named here, IL-2, IL-15, IL-8. Furthermore, each has its unique alpha chain that for IL-15 is really a pivotal partner. They signal through JAK1, JAK3, and STAT5. So for a long time, I thought IL-15 must be IL-2 light. They must be very redundant. And in part that is true. However, they do have really unique roles. IL-2 has a role in preventing immunity to oneself, to prevent autoimmunity, and is pivotally involved in the fitness of regulatory Tregs and in the phenomenon of activation into cell death. In contrast, IL-15 inhibits IL-2-mediated activation into cell death, and its role in terms of long-term protection against invading pathogens stimulates the development of CD8 memory phenotype T cells. Knockouts of these IL-2 and IL-4 alpha versus IL-15 and IL-15 alpha have quite distinct phenotypes as predicted by these different roles. With IL-2 knockouts, there's a massive enlargement of peripheral lymphoid organs as one loses checkpoints, high levels of immunoglobulin subclasses, and autoimmune diseases. In contrast, with IL-15, or the knockout of its private receptor IL-15 or alpha, there is no such lymphoid enlargement, autoimmune disease, or impaired or activation into cell death. Rather, there is marked reduction in the number of NK cells, NK T cells, gamma delta cells, endothelial lymphocytes, memory phenotype CD8 T cells that depend on IL-15 for the maintenance and production. So how can there be difference? There are many differences, but I will only show you one indicated, developed by Sidney Dubois in the laboratory. IL-2 is a secreted molecule that acts on preformed, high-affinity heterodimeric receptors that are on other cells. In contrast, IL-15 is predominantly not secreted, but rather acts as part of an immunological synapse. IL-15 receptor alpha on antigen-presenting cells, indeed IL-15 and IL-15 alpha shown on your left, are made together when the cell is exposed to interferon, CD40 ligation, or toll-like receptors. And these cells with recycling IL-15 and IL-15 alpha circulate for a number of days until they meet with their target, a beta-gamma expressing NK or CD8 T cells that does not express IL-15 or alpha, and transpresents IL-15 signal to the cell for its benefit. When we studied IL-15 due to the differences we just discussed in a considerable number of tumor models. This was the first study that we did in the CT26 tumor model, and one sees a modest effect of IL-15 prolonging the survival of tumor-bearing mice. Studies of these 4 different types of studies such as these, and the indication that it was not really very toxic in rhesus macaques, that the immunotherapy agent workshop, which identified agents that might be of value in the clinic, IL-15 was number 1 in its list of such potential useful immunotherapeutic agents. With that in mind, and with the financial help of our scientific and institute director, we had an opportunity to oversee the biopharmaceutical development program, Dr. Shivambitz and Creekmore, in the production of E. coli IL-15 for human use. And with this, we, with Kevin Conlon, performed the first in-human study of IL-15 in patients with metastatic malignancy. The patients had a much more toxic path than the rhesus macaques. There was fever, shaking chills, hypotension, and one had to de-escalate with the dose of IL-15 to 3 micrograms per kilogram. Nevertheless, as shown here, following the administration of IL-15 by bolus infusion, there was an immediate virtual disappearance of NK cells from the circulation that may be marginated in postcapillary needles or actually enter the tissues. But before Ki-67 new synthesis kicks in, the NK cells began to reappear in the circulation, so that one saw a bolus infusion increase in the number of NK cells. However, we were not satisfied, especially because there were no PRs or CRs that the 5 signal lesions had a 20 to 30% reduction in size. In 2 patients, there was a disappearance of lung lesions. We noted this high Cmax, a high level of IL-15 if it was given by bolus injection. When this was done in rhesus macaques, as shown here, with 20 micrograms per kilogram, the Cmax was 720 nanograms per mil. The reason, if we had a different dosing strategy, we might have lower Cmax and less toxicity, and more efficacy, and that was the case. When we gave IL-15 subcutaneously, the Cmax was lowered by a log, and yet another log by continuous intravenous infusion, where one had a much longer period of time when the recipient had an active dose of IL-15. So, with the Cancer Immunotherapy Network, we did a study of 19 patients with IL-15 in a dose escalation trial. And noted that in the outpatient setting, this is quite well tolerated with a grade 2 pancreatitis as the only serious toxicity. One noted an increase in the number of NK cells, especially in the CD56 bright NK cells. Furthermore, we gave the IL-15 as shown at the base of each of the graphs in light blue, but the maximum response in NK cells appeared after we had stopped giving the sub-Q IL-15. This is more dramatically seen when we administer the Kevin Conlon IL-15 by continuous intravenous infusion to patients with malignancy. Here, the IL-15 was administered during the red arrows, and there was a burst of lymphocytes that appeared at the time, next few days following termination of the infusion. During the rapid proliferation, red cells were not returning to the circulation or were in post-capillary mediums. But anyway, one noted this marked increase in the number of NK cells, 30-fold increase in NK, 350 CD56 bright NK. To remind you that there are 2 types of NK cells, the majority involved in cytotoxicity are CD56 in blue. CD56 bright, the ones most stimulated by IL-15 were there. They had been called immunoregulatory, producing IL-10, and were not felt to be very effective cytotoxic cells. However, Sidney Dubois has demonstrated in green that the injection of IL-15 was associated with an increase in both 56 bright and 56 dim cytotoxicity, and this cytotoxicity included that mediated by NKp46, by ADCC with an anti-CD20 monoclonal, the CD16, and the NKG2D decay resistance. So we are generating cytotoxic cells. So if we're generating this huge number of cytotoxic cells, why are we not getting efficacy? Well, as you know, NK cells of the person are inhibited when they recognize self-class 1 MHC expressed on the tumor. So if it's expressed as HLA-E, NKG2A is the receptor of this negative signal. If it's in class 1A, HLA, then T is for humans. So we're having a turn-off of the system. And although, so that we have to use IL-15 in combination to get efficacy, and we do this by taking advantage of the role of NK cells in the phenomenon of antibody-dependent cell-mediated cytotoxicity killing of tumor cells. And so here is one of many different types of studies. In a fully immunocompetent mouse, EO4, the leukemic cells of the mouse, were transfected with human CD20, and the mice were treated with the anti-CD20 monoclonal antibody rituximab. When we did this in mice, in red, IL-15, very modest effect, rituximab, moderate, but the two together universally cured 80 to 100% of the mice in various studies. In the xenograft model, alemtuzumab, anti-CD52, in a model of HTLV1-associated adult T-cell leukemia, agent focused in the laboratory, we again saw in yellow on the left little effect with IL-15, CAMPAT, anti-CD52, modest, and the two always giving you an additive synergistic effect. If we did these studies in Fc receptor gamma knockout mice, those that cannot do ADCC, all efficacy was lost. So we are looking into what cells involved. Most people believe ADCC is mediated by NK cells, but there's a group headed by Jeff Ravitch and Rockefeller that concludes that no, it is macrophages that is involved in the ADCC killing. So we did our EL4CD20 study, and we eliminated NK cells with HLA-GM1 in red and lost all biological efficacy. But also, when we used chlorinase lysosome, as shown in blue, we lost the majority of activity. It appears one needs both cells acting in combination. And we have shown with deletion studies and with IL-15 that one must have both cells and IL-15, which induces RAY1 or decay for humans and NKG2D, the latter on NK cells. One induces on NK cells more CD16, more Fc receptor gamma 4 needed to bind the monoclonal. So we are augmenting with IL-15 both the number and the functional capacity of NK cells. With these in mind, we're initiating a series of studies where we're using anti-cancer monoclonal antibodies in association with IL-15. Some of these are in adult T-cell leukemia lymphoma, which is an aggressive disorder of human CD3, CD4, and CD25 cells, which we in 1984 showed to be profound immunosuppressants. They are leukemias of Tregs. And these, there is no curative therapy other than bone marrow transplant. So when in the past we studied alemtuzumab, anti-CD52, we noted that 12 or 15 patients with the acute leukemic form of this disease had a partial or complete response, and 2 complete chronic. However, the responses were of short duration. We wish to increase the proportion of responders, especially the duration, by adding IL-15 to augment the ADCC of alemtuzumab. And so we start by treating in a dose escalation form with IL-15, followed by alemtuzumab in a conventional dosing strategy. We've only treated 2 patients. The first one with the acute form of ATL. I show here where in blue are the number of cells, lymphocytes in the circulation, and in red, IL-2 alpha, a mediator, measure, a surrogate marker of tumor activity. What thus is having in the periphery a good response. But we also note the limitation of this approach. The antibody does not pass the blood-brain barrier, and thus this patient has developed neurological adult T-cell leukemia, which for the future we will have to evaluate that beforehand and use intrathecal methotrexate. But anyway, this is one. We are approaching an effort to use adalimumab, an anti-PD-L1 antibody that does ADCC, in the treatment of T-cell malignancy and one form of solid tumor. And similarly, uses today's approach, IL-15 with an anti-cancer monoclonal antibody, with the anti-CD20 obinutuzumab for CLL. So we are using a refractory relapsed CLL anti-CD20 monoclonal antibody plus IL-15. It is hoped that we can thus take advantage of the effect of IL-15 on macrophages and especially NK cells with this approach. However, IL-15 always has a partner, IL-15 or alpha. Indeed, there are those who consider these a heterodimeric cytokine. What was noted by others is that they must be together in mice to generate effective IL-15. If one has an animal with bone marrow transplants of cells that express only IL-15, and in the same animal, only IL-15 or alpha, as shown on your left, well, does not generate effective IL-15, and the mouse has no NK cells which depend on IL-15 for their production. In contrast, if they're produced by the same cell as shown on the right, you have IL-15 effectively produced, transpresented, and NK cells developed and are maintained. In addition, there were studies of cocoa where normal NK cells were injected into wild-type mice in blue or to mice missing IL-15 or alpha. And as you know, in wild-type mice, there's a meaningful survival, but virtually no survival in mice who do not have IL-15 or alpha. Thus, one is moving to situations where one will administer non-IL-15